do. Hi everybody. Just going to wait a few minutes for people to roll up, and then we can begin. I don't know. How about I wasn't making any noise? Can you hear me now? Blah 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 blah. Sorry, my typing sucks at the moment. Any better? Let's see if I need to up the juice. Oh, cool. Hey, I mean that'd be kind of disastrous, right? If you can't hear what I'm saying. Don't think you'd be able to lip read a genetics lecture. All right, well, might as well get cracking. As people are rolling in, they can always look back at the YouTube video if they need to see whatever they might have missed in the first few minutes. Okay, so what we're going to do today is we've got two. Hey Tanya, yeah, we've got two PowerPoints to go through. To be honest, the first one is fairly light. It's really just a bit of a kind of an overview, you know, some terms and stuff which are important to have, but really it's nothing that you need to worry too much about. So I'm just going to hit the couple of things which are particularly important in that, and then use that to free up the time for the rest of the lecture to go through the flow of information PowerPoint, pardon me, which is the second one in the list. That's got a lot more kind of basic information in there that you need to know. And really that second PowerPoint, that flow of genetic information PowerPoint, is really in many ways a recap of what you have already learned in GenBio 1. Right, so there shouldn't really be anything that's completely new there, but there will probably be things which you might have, you know, be a little rusty in, essentially, depending on how long it's been since you took GenBio 1. Right, obviously it doesn't hurt to refresh your memory, and also a lot of that stuff is super important to know and understand. Okay, so let's get going. Doopy doopy doo. Yeah, we don't want that one. We want this one. Right, so really it does actually pay to kind of define what genetics is. And really, because you can talk about it around a lot of different ways, right? And you can go on, kind of skirt the issue to some extent too. But really it's the study of inherited traits, right? And so traits are, you know, essentially phenotypes which are encoded by a genetic locus, right? And that's a lot of locust, right? So there's no T at the end of that. It's essentially a position on your genome which has one or more genes in it which encode a particular trait. You know, could be, you know, sickle cell disease, for example, right? So that's encoded by a beta, I think, hemoglobin gene, right? So it's either functioning or it's not functioning. and that the trait is essentially normal cells, red blood cells, and you either have the normal phenotype or the sickle cell phenotype, right? So, and then essentially, how is that trait inherited, right? What mode of transmission does it have, essentially? If we're looking at, and that's looking at individuals in family trees over time, essentially, right? Also, and it's kind of self-evident, really, right? If you're trying to study a trait, let's say sickle cell disease, right? You want to know, well, what causes that trait? I mean, it's all well and good knowing that if both of your parents have sickle cell disease, then it's quite likely that you'll have sickle cell disease, right? In fact, it's 100% likely. But what causes that, right? You need to understand the basis for that trait, whatever it is, and it doesn't necessarily have to be a disease trait, right? Could be all kinds of different things, right? People have spent many, 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 many years trying to figure out the genetic basis of intelligence, right? For both good reasons, to help people that are suffering from mental retardation, perhaps, and also for really, really dodgy reasons, like selecting for like super brainy people, which is eugenics, right? We don't want any of that. But so we really want to know why, right? And so that's really the study of the hereditary material, right? And really what we're talking about here is DNA sequence information, the genes that encode a particular protein, the protein that they encode, what does it do? And also, how do we get from one to the other, right? How do we get from DNA to protein or RNA if RNA is the active molecule in case, right? So without knowing any of that information, the basis for that trait is largely a mystery, right? We want to know because we're curious monkeys, right? We really want to know why something works the way it works. At least I do. That's kind of why I'm a geneticist, essentially. And so that's really what genetics boils down to, right? What's the basis for a particular trait, molecularly and biochemically and in terms of cell biology or whatever, in terms of its actual mechanism, right? And how is that trait inherited? How does it pass from one generation to another? And how does that trait change within a population over time? Right. So the cool thing about genetics is that we're really working on a large range in scales. Right. We're working on the individual bases that make up a DNA, make up a gene, right? All the way through to events that span hundreds, if not thousands, and potentially millions of years, right, to bring us what we have now, right? And there's all sorts of really cool examples about that. So really, in terms of kind of fields of study or divisions within genetics, right, these are all like semi-artificial divisions, right? Because, scientists like cutting things up into nice little chunks, makes it a little easier to understand and grasp. But by and large, genetics kind of falls within kind of three main areas, right? Which is transmission genetics, study of inheritance, essentially. Molecular genetics, which is really kind of more what I do, which is essentially the what is that material made up of, right? And how does it get used, right? So that's all about, DNA sequence, both within an organism, between species and that sort of stuff. How that DNA sequence is used, copied, mutated, stuff like that. Or RNA sequence as well, if you're looking at RNA viruses. And then we've got like the big kind of like long-term picture genetics, which is evolutionary genetics. And that's really the key factor there is time. It's really the main differentiator between evolution genetics and everything else is that they really care, well, people that study it really care about time. How does this particular um, trait or allele and its frequency change in a population over time? So in a sense, you can also think about this in terms of scale as well, right? If you're looking at molecular genetics, you're really kind of interested in what lies within a cell. You know, what DNA do we have in that particular nucleus? How is it getting transcribed under what conditions, and so on and so forth, right? We're kind of working at, like, well, actually, you can't see the gap between my fingers. Where's my webcam? There you go. We're talking about that kind of scale, right? Transmission genetics kind of ups it to the organism level, right? So really, in terms of transmission genetics, it's about what collection of alleles do I have, and which of those did I pass on to my children, and what alleles did I get from my mother and from my father, right? So really, when we talk about transmission genetics, we talk about individuals, right? And probabilities, right? That's where the math comes in a lot of times in terms of genetics. And then obviously evolutionary genetics, we're talking about populations. So we're no longer talking about, me and my family, or maybe my extended family, or maybe even my neighbors. We're talking about San Antonio, or we're talking about white settlers in a particular country, or Polynesian settlers in a particular island, where we're talking about populations and how the makeup, genetically speaking, of those populations changes over time, right? So obviously these are all kind of smooshed together in different ways, right? It's very rare to find someone who's just one thing, or interested in one aspect and not another, right? So, for example, in my work, it's almost entirely molecular genetics, but I do a lot of kind of transmission genetics to be able to study those molecular genetics, right? So, but I don't do any evolutionary genetics at all. Well, I don't now. Used to, So you see, it kind of depends on the question in hand, right? What are you asking, and that determines how you ask that question, right? So that's really. Oh, and also in this course, we're really going to be focusing on the first two, right? Transmission genetics and molecular genetics. We're not going to be touching really at all on evolutionary genetics. That's what you will study in evolution, right? So the uh, evolutionary and population genetics is kind of split off, and that's put into the evolution class. So really, genetics and evolution form like pardon me, a paired course, right? Where in this course you learn the small scale stuff, molecular and hereditary genetics. In evolution, you learn the big scale stuff, population and evolutionary genetics. Right, so that's just how we split it up here. There's not really a whole bunch else in here that's super important, because we're going to cover it anyway later on. Just to introduce the term genomics, right? So that's the other point of note, right? Most of you, most likely know or already heard of this, but it's really about studying not just one or two traits. It's about studying the entire genetic information of an organism, right? And potentially comparing that to other organisms or other species, right? So then we start getting into kind of a more evolutionary flavor of genetics, right? And so really this is something that has come about with advancements in technology, right? So the first, oh, when was it? First genome was sequenced in, I think, 97 or 98, actually probably before some of you were born, which is kind of crazy, which was E. coli, done the old school, really hard way, using like gels and stuff. The first eukaryote, as in animal, sequence was C. elegans, and that sequence was published in 2000, right? So 20 odd years ago. Then humans came, I think 1 or 2 years later. I can't actually remember the date, right? And so that was an enormous effort. I think in today's money, that sequencing the human genome cost, I think it was a couple of $1 billion, right? So that's like 1 or 2 followed by a whole load of zeros that I'm never going to see in my lifetime, right? So that was an enormous cost. Right now, today, you can get your genome sequenced for around $1,000 to $3,000, right? So that now becomes, I mean, that's like the cost of an MRI, right? Which is a fairly standard medical procedure, right? Which insurers cover. So now, and actually, we've had this happen within my wife's family. Now, if you get cancer, now your doctors or your cancer specialists now can potentially sequence your genome to try and figure out what the genetic basis of that cancer may be, right? And in doing so, and that's data that has been culled from, you know, decades of genetic studies of individual cancers, right? So developing markers essentially for a particular type of cancer, right? And so now you can look for those markers, right? But you can look for all of them, right? You can't just go, well, that's a cancer of the lung, so I'm going to look at this gene, right? Because I know that that often causes cancer in the lungs, right? Now you just sequence a patient's entire genome, and that gives you answers which you didn't have access to before, simply because of that decreased cost of sequencing, right? And really, basically, that's just advancements in technology, right? Kind of the same difference that has gone from uh, something like that, right, to the supercomputers you have in your pockets, right? I'm a bit of a Luddite, so I don't have one. So anyway, that's really opened up a whole bunch of stuff. Now we can do some really kind of broad scale comparisons, right? So that's another kind of question of scale. Right now we're not talking about, 1 or 2 traits and how those are inherited. Now we can talk about everything and see how that changes over time, which is also pretty cool. And also, as I was talking about the medical stuff, it's opened up a whole world of treatment, and it's actually now called personal genomics, right? So figuring out what a patient's genome is actually opens up a lot of different treatment options, right? You don't have to guess. Like for example, sorry, I'm kind of going on one of my tangents, right? This happens every so often. There are lots of different genes whose, depending on what alleles you have, right, depending on what variants of those genes, right, because we all have the exact same genes, right, but we just have different versions of those genes or different like small changes in them, right? So for some people, if you give them a particular chemotherapy drug, right, they have particular alleles which destroys that drug very quickly, right? Or pumps it out, and then you kind of get rid of it through your liver and your kidneys, right? You basically pee it out very, very quickly. So if you were to use that particular drug, the patient wouldn't respond because or you'd need a much higher dose of that drug, which may then cause other problems, right? Flip side, on the other hand, there are people with the exact same cancer who have a variant of that gene, an allele, which means that they don't metabolize that drug at all, right? So that drug can then build up to a much higher level and actually cause serious side effects and toxicity, right? So before, in terms of cancer treatment, it was a kind of a guessing game, right? Well, we're going to give you this drug and see if it works, and if it doesn't kill you, then yay, that's great. But did it kill the cancer? No, it didn't. Oh, we'll try this drug, right? So it took a lot of trial and error to find the right treatment course. Now it's possible just to sequence a patient's or genome and figure it out from the get-go, right? So that's incredibly exciting time in medical genetics. That's kind of it from that. Tanya?
Professor Crook,
Mhm.
so how much data would be in a personal genome since there's a lot of like variations and
Yeah, so that's a tricky question because we haven't, well, essentially only, I think like 5% of our genome codes for protein coding genes. And then an unknown amount encodes for non-coding genes that produce mRNAs that do stuff, and the rest is just kind of ancestral cruft, basically. So there's a huge amount of variation in areas that don't matter, and actually a surprisingly little amount of variation in areas that do matter, right? So it's sometimes a case for seeing the wood for the trees, right? And that's where people who specialize in this really bring a lot to the table because they know what they're looking for. So really, in a sense, what you're doing is you're not, well, you're sequencing an entire genome, right? Which is, what's that, 6 billion bases, I think, off the top of my head, right? So gigabytes of data, basically. But really, when you're using that as a tool, what you'll do is you'll have a probably like a list of, I don't know, say 100 or 200 genes which are known to affect cell cycle control, metabolism, what else? DNA repair, DNA checkpoint proteins, things like that. And then you'll look to see what variations or variants or alleles that patient has for those. All the other stuff, things that are involved in limb development, you're not going to care about because you're looking at lung cancer, for example. Pardon me. So yeah, it's kind of like you get all the information, then you have to drill down in a targeted way to actually figure out what it is that you're dealing with. And that in turn builds on kind of the molecular genetics that I was talking about earlier, which is essentially decades of, hordes of labs studying individual genes and their roles in that particular type of cancer or drug metabolism or whatever, right? So in many sense, science is a pyramid, right? You may have that kind of amazing discovery at the very, very top, which cures a particular cancer, or cures a particular patient, right? But that's built on this kind of huge base of information that kind of leads to that point. So it doesn't really matter where you're on that pyramid, you're still contributing to the overall goal at the end of the day. Cool. Does anybody have any questions about any of this stuff? It's just kind of basic terminology, really. All right. Okay, so now this is where we get into the kind of nuts and bolts, essentially. Right, so there's kind of different ways of looking about this. This is really a way of kind of starting at ground zero and leading up to Mendelian genetics, which is really the study of transmission of traits, right, inheritance. And really you need to start with the very, very like smallest units of information first. And in terms of life as we know it, that is DNA nucleotides, right? So deoxyribonucleotides. If you're talking about RNA viruses, which are like this awkward kind of like, oh, right, there's RNA viruses over here, then obviously those are just ribonucleotides, right? But the building blocks, like the letters, right, essentially that make up the book that is our genome, right? So I quite often use the analogy that DNTPs, like the A, T, C's, and G's, they're really the letters, right? The words are kind of, I don't know, they're codons, essentially, right? We'll talk about those later. Sorry, puppies are happening. And, individual, I don't know, paragraphs, I guess, would be genes, right? So that's kind of like the analogy that I like to use, right? Really, your genome is like a book, right? Individual chromosomes, they're chapters, right? Individual paragraphs, those are genes. Individual words, those are codons. Individual letters, those are your A, T, C's, and G's, right? And so just in the same way that we use a limited set of characters, depending on which language you speak and use, right? We have, what is it, 26, I think, 24? I can never remember, actually. Someone will correct me if I'm wrong. We have a limited set of letters or characters that we use, and we have this huge vocabulary, right? Essentially. right? We have this, and huge body of literature, right? And that's essentially the same with DNA. Same idea, right? It's information as well, right? Uh, no books have ever baked a muffin, right? It's kind of one way of putting it, right? And in the same way, DNA doesn't do anything. It sits there. It's a repository of information, right? And so uh, we'll get to how that information is used later on, right? In terms of gene expression and control of gene regulation, essentially, right? So again, this is like a really fairly straightforward recap of stuff that you either should already know or did know and you need kind of a refresher on. But essentially what we need to know about DNA is uh, kind of how is it made up, right? What are its properties and what's its structure, right? And all of those three things are really important for what we study later on, right? Because we need to understand its makeup and how it's made, because we're going to be studying DNA replication and transcription, right, which are super important. Its structure is, oh, actually, I shouldn't use that finger, sorry. Its structure is super important because we've got to think about that in terms of packaging, right? And so we'll get to that later on when we kind of start talking about the conundrum of how DNA is actually packaged into a nucleus, right? Which has bigger ramifications in terms of how that DNA is actually accessed as well. So the key points that you need to know about DNA is obviously it's made up of 4 bases, right? We have, actually, let's get these down as adenine, guanine, cytosine. Apologies for spelling mistakes. And thymine. Oops. Typing without your index finger is a real pain. Right, so those are the 4 bases. Anybody remember what's different about RNA? Quick question, so you can type it in fastest. What changes in RNA? 2 things.
T for uracil.
Right, so we've got uracil. What's the other big change between a DNA nucleotide and a RNA nucleotide?
The sugar.
Yeah, what about it?
It doesn't have deoxy. It's not deoxyribonucleic. It's just the ribo.
Okay, what does that mean though? You're right, very much so.
I just know it's a different sugar.
No, you're totally correct. So kind of let's roll that back a little bit. What's attached to the bottom of the sugar? Actually, I think you can see that in the PowerPoint. I'll bring it back up again.
I think I don't remember which one it is, but I know one has an OH connected to it, while the other one just has an H.
That's a good point. Well, DNA has one OH, right? What does RNA have attached to that sugar? So if DNA is deoxy and it has one OH, just a priori without knowing anything else, what does RNA have?
It's only one. It's only one hydroxyl group.
2.
2.
Yeah. So DNA has one OH. That's not an O. That's an OH. Sorry. There you go. What carbon is that attached to on the sugar? What's the terminology we use for direction in DNA and RNA? Yeah, I'll give you a clue.
Helical?
In Transformers, we have Optimus.
It's prime whenever they like 3 prime, 2 prime, 1 prime.
Yeah, so we've got 5 prime for one end and 3 prime for the other end, right? So that 3 prime is essentially the 3rd carbon in that sugar, right? And that's where the one OH group of RNA is attached. Okay, get to sorry, DNA. My apologies. And let's actually put RNA has 2. One on, oops. Hang on. Right, so that's why, Tanya, DNA is deoxy, because it has lost the OH group on the 2' carbon. It's just an H, as someone said already, right? So what is the 3' OH used for? Actually, this ties back into a question you asked me in lab yesterday. What's the importance of that 3' OH group that's dangling at the bottom of that sugar moiety?
It's used to connect to the 5' H from another DNA.
Yay, totally, exactly. Super important, right? If you do not have that, you get no more extension of the DNA or RNA strand. It's also the same in RNA too, right? So that 3' OH is essential for the polymerization of DNA and RNA, right? The dehydration condensation reaction that kind of forms that phosphodiester bond between the 2 bases. Actually, another short tangent, in what's called Sanger sequencing, right, which is how the human genome is sequenced, is how your yellow fly genes will be sequenced later on in the semester in the lab. That actually uses special nucleotides called dideoxy nucleotides. They actually have that 3' OH removed. And so if you add a small amount of those into a PCR reaction, you'll essentially stop the extension of that product wherever you find that base, right? So if you use dideoxy ATP, right, which is adenine, then every time you have an A, there's a chance that that product will be stopped, right? You'll stop that DNA extension. And that's actually how you sequence DNA by the Sanger sequencing method, right? So that's kind of like a manipulating biology to kind of do something that we want it to do. Anyway, that's a bit of a tangent. Right, so you see how these things are really important, right? These are not just labels, these are also really, they tie into how DNA replication occurs, right? And that also, these 5 to 3' names, that also gives us a direction, right? So DNA, RNA, and as a consequence, proteins have a direction, right? Even though they are linear molecules, right? They kind of go along in a sense in a 2-dimensional way, right? In their simplest form. DNA, RNA, and proteins have direction. Starch does not, right? So starch is a much simpler polymer, and it can't tell one end of a starch molecule from another end, right? It's pretty much exactly the same. But DNA, RNA, and proteins do, right? So extension can only occur at the bottom of that chain at the 3' end. So you cannot add a new nucleotide, whether DNA or RNA, to the 5' end of a chain. You can only add it to the 3' end. Right. And the bonds that link these together are called phosphodiester bonds. Phospho because it's on either side of a phosphate, ester because it involves oxygen. It's one way of remembering it. Okay. So moving on to here. Right, so we already covered this. Couple of other things which are of different degrees of importance, right? Obviously, DNA is double-stranded. We know that. that's actually one of the reasons why it's so durable, right? One of kind of two reasons, actually. DNA, as you know, is like super tough, right? I think the oldest organism that we found it from is about 60,000 years ago, give or take. Actually, that's the whole basis of Jurassic Park, right? The series of films. So DNA is really, really robust, which is kind of what you want in terms of an information storage molecule, right? You don't want it falling to pieces under kind of stress or hardship, right? And so part of the reason why it's so tough is it has these vulnerable reactive bases inside, right? So we have the double strand, right? It's a right-handed unequal helix, right? Which we'll get to in a second. And the kind of the information is stored on the inside, right? So those bases pair by actually, can anybody remember? This is another throwback to GenBio 1. What kind of bonds hold the bases together? The A's and
Hydrogen.
T's. Well, that wasn't too hard, right? Hydrogen bonds. Okay, then answer this. Why hydrogen bonds? What makes hydrogen bonds the perfect ones to hold together the two opposing strands of DNA?
Their cohesive properties.
So how do those rate compared to, let's say, a covalent bond?
They're a lot weaker, so it's easier to break them apart and like connect them back together.
Yeah, exactly. That's really good. Sorry, I didn't see what someone said in chat. Motaz, actually, they're one of the weaker bonds, to be honest. Hydrogen bonds are what hold water molecules together, right? And so when you boil water, what you're seeing is those bonds being broken and water molecules kind of flown around. That's what essentially steam is. And so was that Gabriel? Sorry, I didn't catch who was speaking then.
Yeah, it was me.
Yeah, so that's exactly the reason is that hydrogen bonds, when you have enough of them, right, are strong enough to hold DNA together, right? If you think about like water boatmen that kind of run around on the top of the surface of water, that's enough strength to kind of keep those insects from, going in the water. But they're also weak enough that you can split them apart. And one of the things that we need to do regularly, depending on conditions and what cell we're looking at and so on and so forth, is we need to open that DNA, right? Because the information's on the inside, right? So if we want to either read that information in terms of transcribing a gene and doing something with that information, or if we want to copy it, we have to pull that DNA apart, right? And if those are covalent bonds, or actually ionic bonds would be pretty shitty too, because they would be affected by the availability of water, right? Which, we have a lot of, right? So if those are covalent bonds, it would take a hell of a lot of energy to break those bonds apart and then reform them. Whereas really, if you think about my favorite analogy for DNA is a zipper, right? So essentially it's something that holds together pretty robustly, but you can open it up at will, whenever you need to, right? So these bases are held together by hydrogen bonds, and the number of hydrogen bonds and their orientation, because really bases are held together like that, right? They're not going in the same direction, they're going in opposite directions, right? That's the anti-parallel nature, which we'll get to in a second. But these bases fit together based on how many hydrogen bonds and also the orientation and shape of those bases, right? This is possibly one of the most important properties of DNA, right? Because this is what allows you to copy it with extraordinarily high fidelity, right? That's why, over the thousands of cell divisions that we undergo to generate us as an adult, or, something along the way to being an adult, right? Our DNA is largely the same at the end of that process as it is at the very beginning when our zygote was fertilized, right? It's also why things like HIV and influenza and coronavirus, right, mutate so readily is because they're made up of RNA, right? And so the copying of RNA is much less faithful than that of DNA. Okay, that's a little bit of a tangent, but essentially what I'm getting at is that A's and T's bond, so adenines and thymines, snap together really nicely based on their shape and the number of hydrogen bonds that snap them together, essentially, like kind of number of magnets is one way of thinking about it. Anyone remember how many bonds join guanine and cytosine together?
Isn't it 3?
Yay, that's right. Thank you, Motaz. You got that too. Sorry, I'm a little bit slow on hitting the chat. I'll see if I can stick that down there so I can get back to it more easily. Yeah, it's 3. So where would you, here's another question, kind of getting you thinking about properties of DNA. In any given region of DNA, let's say, talk about eukaryotic chromosome, there are multiple, what we call origins of replication, where DNA replication begins, right? What would you expect to find more of in terms of bases in that region? Would you expect to find more A's and T's or more G's and C's?
More A's and T's because they're an easier bond to put together because there's only 2 combining
Right.
them.
And they're easier to break apart. That's also Motaz and Rose got that as well, right? So if you want to have easily accessible areas, you find these in the promoters of genes as well, where you're going to be pulling apart the DNA to be able to copy, transcribe them with RNA polymerase. Another question, which will kind of put this more in the context of kind of evolutionary biology, I guess. If you were to dig up a hyperthermophile from some kind of black smoker in the Pacific, right, 4,000 meters depth, probably they're happy living at about 105 degrees Celsius. What would you expect their GC content to be of their genome relative to a bacteria that you find, you know, trundling around in our guts?
It'd probably be higher.
Right, exactly. So you can actually use, so this actually reflects in many ways the environment in which that organism has evolved as well. Right. So it's just kind of to put this into a bigger context, I guess. Right. This is stuff which has, it's not just kind of facts to remember, but they're really kind of foundational. See you later. Foundational properties of an organism, right? Which kind of, you know, natural selection has acted upon over many, many years. So the last thing that I want to hit on is this anti-parallel word here, right? And so this also has kind of important connotations because essentially what it's saying, it's like having 2 railway tracks, right? Two tracks going in different directions, right? One strand be, let's say, well, actually we do it this way around, 5 prime, 3 prime, the other strand, 5 prime, 3 prime, right? These strands have directionality, and that directionality is opposite to each other, right? Not entirely sure why that is the case, to be honest. I don't know if there's kind of some thermodynamic structural property or if it's just an accident of evolution. I really don't know. Or maybe it's just that's the only way that those adenines and thymines will fit together is another possible explanation, right? But whatever the cause of it, it means that you can have information going this way and information going this way. And actually, in rare cases, you can have that information overlapping, right? So you can have genes on this strand go in that direction. You can also have genes on this strand going in that direction. And sometimes they overlap or share the same control regions as well. It also causes all kinds of problems in DNA replication, which we'll get to later. Actually, some fairly major ones. Let's stick this over here. All right, any questions about any of that stuff? before we move on? Oh, crikey, we've got like 30 minutes left. I need to stop talking less and teaching more. All right. So essentially, in terms of flow of information, DNA is a static, well, it's not a static molecule. Sorry, I've got to be careful about making general statements. DNA is a very dynamic molecule, but it doesn't do anything other than store information, right? So if we want to use that information to do something, bake a cake, for example, we need to get that information out of that DNA and into a useful form. And typically, when we talk about useful form, we're talking about proteins, but not always, because actually there are an awful lot of RNAs which do really, really cool stuff too. And there's actually RNAs which are enzymes. They're called ribozymes. Ooh, can anybody think of one? Let's see if this is kind of a little bit more of a challenging question. Anybody think of an RNA molecule that is also an enzyme? This is probably like, what? Enzymes? No, it's true. Possibly the answer to the origin of life, no less. A molecule that can both store information and do stuff.
Oh, is it a ribosome?
Yes, ribosomal RNA, right? It's actually the thing that forms the peptide bonds between amino acids, not the protein. That just kind of provides the context, right? So we're either talking about RNAs or generally speaking, we're talking about proteins, right? And so really that's what this is actually the sequence hypothesis. This is kind of, I need to change that. Where we go from DNA to RNAs via the process of transcription, and then using a ribosome and ribosomal RNA and tRNA, we then translate that into protein. Right, so transcription is really, it's a very good word for this process because we're not changing the form, the content of the information, right? We're changing the form in which that information is in, right? So in terms of DNA, right, it's encoded in 2 strands of DNA. In terms of mRNA, that's a very different kind of type of nucleic acid, right? It has a very different purpose. And it is also able to leave the nucleus, right, which is a key part of this because transcription occurs in the nucleus, translation occurs in the cytoplasm, right? So, but when we turn that into protein, we're really translating that that information from one form into something else. Like it's kind of like a Rosetta Stone in a way. And I'm just going to bounce ahead here. And this slide here is a little bit of a scary looking slide, but it's not as troubling as one might think. This is really the Rosetta Stone of molecular biology. Actually, another quick question. This is kind of a fun one. Does anybody know what the Rosetta Stone is? Right, we use that a lot, like in just general parlance and the like. But what does anybody know what the Rosetta Stone was?
I believe it was a stone tablet, but I don't remember who it was made by. But it was under the translate between like 2 languages that were nearby.
That's right. It was a like as you said, a stone tablet that was used to translate hieroglyphics into, I think, Sumerian, which was an ancient language from the Eastern Mediterranean, I think, around there, give or take. Because before then, no one understood hieroglyphics, right? They didn't make any sense. There was no kind of analogous language that they could be compared to. And so the Rosetta Stone provided that kind of link between those 2 languages, and people understood Sumerian because it was more similar to modern day languages. So there you go. So this is essentially the Rosetta Stone of molecular biology, where we translate these words, right? So these 3-letter words, these are mRNA words because they've got U's in. These words, they're called codons, is their formal name. These are then translated using tRNAs into amino acids, right? And again, we'll get into the mechanics of that in far more detail later on in the course. But this is essentially how we go from one to the other. Anyway, so yeah, that's this process of translation here really uses, I mean, the RNA is, our RNA is the enzyme that does the work. The tRNAs are the translators. The T stands for transfer, but you could also think of it as being translation as well. These are what translate those words into amino acids. Okay, so love a little bit of kind of refresher information. When we talk about transcription, really what we're talking about is a process, right? That basically begins with the recognizing of a bit of DNA that needs to be expressed, right? So we have a particular gene. Let's say you know, you've had a glass of wine or something like that, and your liver goes, "Oh shit, alcohol," right? We need to get rid of that because it's a toxin. And so your liver cells will start producing, will need to start expressing a gene called alcohol dehydrogenase, right? Which basically turns ethanol into something that's less toxic, right? So that gene will have regulatory sequences, right, which will recognize it, will bind to it, and then will initiate the process of transcription, which is, you know, a whole bunch of enzymes doing, essentially they're going to make a copy of mRNA, and you can see it here, this one down here. You can also tell because it has U's in it, and it's going in, it also has its own direction, right, 5 prime to 3 prime. And because this is an anti-paralleled kind of deal, right, we're going to be, the actual gene is in this side, right? This is the coding strand, the one that goes 5 prime 3 prime, because essentially what you'll get is, this is actually a really good, let's see if I can make this a bit bigger. Yeah, you can see a little bit better now, so you don't have to squint so bad when looking at your computer, right? So this is actually a really cool diagram because this encodes a lot of information, right? So this strand here of the double helix is the coding strand. This is the strand that contains the gene or the information that encodes whatever is needed, right? And you can see that because the mRNA transcript is a copy of that coding strand, right? So you have a U here, that's a copy of a T here. We've got an A, well, you can't see the G. You know, you see what I mean, right? So this is a, this mRNA transcript is a copy. Obviously it has U's instead of T's. Now it has to be made in a particular direction, and as a result, it has to use the other strand. This is called the template strand, the one that's being transcribed, to extend that mRNA transcript, right? Again, we'll get back to that more in the future. And as with DNA, the mRNA transcript can only grow at the 3' end, right? So the gene is going in this direction from the bottom up to the top of the screen. The mRNA transcript is being extended from the bottom up to the top of the stream, right? So here's the start of the mRNA. Here's the growing end around here. And obviously, as you can see, that double helix has to be opened up to enable access to that coding strand, that template strand, sorry. Okay, so I tend to bounce around a little bit in these PowerPoints when I'm talking, because it kind of, teaching isn't necessarily a linear process. So hopefully I don't lose you in doing so. Right, and so when we get to translation, right, that transcript, and so again, keep thinking about this in terms of flow of information, right? The information is stored in DNA, right? Doesn't do anything there. It's transcribed into mRNA, and that information, that kind of unit of information, I guess, that is transported out of the nucleus into the cytoplasm, where it's now used as a template to translate that information into protein, right? And so this is what I was talking about earlier, right? Every 3 bases of the transcript are words, they're codons, right? And so each of these codons, these 3-letter words, this is AUC, which is the start codon, codes a methionine. It's at the beginning of every, as far as I'm aware, every mRNA transcript, or the codon region of every mRNA transcript, rather, of every organism that we know of, right? So this is the start codon. It's a AUG, and it encodes methionine, right? So that's the first amino acid of every protein. Doesn't necessarily mean it's going to be the first amino acid of every mature protein, but as it's being translated, that's the very first one. Anyway, so these, remember I was talking about tRNAs being like the translators, right? And you can see how here. So this tRNA has a particular structure, and it also has a particular sequence. This is called an anticodon at the end of it. And based on the same principles that make A's and T's and C's and G's stick together, right? Hydrogen bonds and shape. The anticodon base pairs with the codon. So an A in the mRNA will base pair with a U in the anticodon in the tRNA, U and A, G and C, and so on and so forth, right? So because A's can't bond with A's, or C's can't bond with T's, or anything like that, right? They have a very specific base pairing. Because of that, this particular codon, which carries this particular amino acid, will only stick to this particular codon in the mRNA, right? That's the basis of translation, right? These tRNAs will only recognize one single codon, and they can only carry one single amino acid, right? And so because of that specificity, you'll only ever get methionine in the same kind of, position in the transcript as you will find an AUG, right? And that's really where this codon table comes in, right? Now, one thing that is kind of funky, and this is pretty interesting as well from an evolutionary point of view, right, is that you can have, so only one codon can only encode one amino acid, right? It's utterly specific like that, right? You can't have one codon encoding 2 amino acids, right? Because it just doesn't work, right? That's why we have these tRNAs, right? However, one amino acid can be encoded by more than one codon, right? So you see here, I think leucine is the champion, right, in terms of the numbers. Leucine, the amino acid, can be found in or put in place where there are 6 different codons, right? So if you have a CUU, you have a leucine. If you have a CUG, you have a leucine. If you have a UUG, you have a leucine as well, right? So this is called degeneracy, right? So the genetic code is said to be degenerate, which I always think is kind of a funny way of describing it, to be honest. And this is actually really kind of useful. It's actually a very important buffer against mutation, because you can see that, say, for example, in leucine, you can have the last letter of that codon be anything, and it doesn't matter. You'll still get a leucine in that place, right? So if that leucine is a particularly important amino acid in that protein, for whatever reason, like enzymatic active site or DNA binding or whatever, then you have a lot of robustness against the effects of mutation, right? Because you can have mutation occur and in that protein sequence, and it may not necessarily change the amino acid sequence, or sorry, in the DNA sequence, but it won't change the amino acid sequence, right? So this degeneracy is kind of a form of robustness against mutation. There are a few codons which are kind of single purpose codons in a sense. One of them is methionine, right? So this is the start codon, and only an AUG will give you a methionine, right? And I think there's actually another one. Tryptophan is another one, which is kind of weird. Don't know why that one is, but we won't worry about that. And actually, what you'll find is this is the codon that's mutated in the yellow fly gene that we'll be working on in lab, right? So this is why yellow flies are yellow, right? Because the start codon for their yellow gene is broken, so you don't get any protein. There are also 3 other funky ones. These are called stop codons, right? So these are UAA, UAG, and UGA, or termination codons, right? And essentially, when an mRNA transcript is being translated, you have to stop it at some point, right? Because you don't want to just kind of keep going until the very end. You want to only produce the protein that you need to produce. And so every coding sequence, right, the bit that encodes a protein in an mRNA transcript has a stop codon at the end of it. And there are no tRNAs for stops, right? So as soon as the ribosome gets to that point, it pauses, and basically another protein comes in and, you know, turfs it off of the mRNA. And so translation is stopped, right? So these methionine and the stop codons are essentially bookends of the translation process. One's the beginning, the other 3 are the end. All right. Anybody got any questions about any of that stuff? Time flies when you're having fun, right?
I had a question, but I don't know if it's more specific than what we're talking about.
Yeah, fire away.
I was curious about because I've been looking into the Huntington's disease, and I
Oh,
was
yeah.
wondering how
Oh,
because it's caused by the CAG repetitions.
polyglutamine disease.
Yeah.
Oh, yeah, there we go. Uh-huh.
I was curious of how the repetitions fit into that.
Ooh, well, that's absolutely a fabulous question. Sorry, what was your name? I can't see the user list up.
It's Rose.
Rose. That is an awesome question. So that's called a poly-Q disease or polyglutamine disease, and it's actually part of a larger group of disease called a trinucleotide repeat disease. And so essentially what you have in I can't remember the name of the gene, Huntington, I think, or something like that. You have a stretch of repeat codons, right? So rather than having a leucine and then a serine and then a threonine and whatever, you have a series of CAG repeats, right? And they do encode amino acids, right? So they have a function. Pardon me. And at a certain number of repeats, you don't have a disease, right? That's why Huntington's tends to appear much later in life, because it's essentially an expansion of those. But what happens is when you copy your DNA, DNA polymerase doesn't do very well with repetitive DNA. It does what's called a stutter. And so when DNA polymerase hits that kind of series of CAG repeats, it quite often pauses and like stutters. And so it will add another CAG repeat. I'm not entirely sure the mechanism for that, to be honest. I'm kind of using a fairly like wiffly waffly term. But the end result is that when that cell divides, the daughter cells will typically have an extra CAG repeat. right? And so you can imagine that over time, as we develop as organisms, we go through, hundreds and thousands of rounds of cell division to get to the millions and billions of cells that, we're made of. And so every time that expansion increases, right? And above a certain number of repeats, that causes the protein to fold improperly, and it causes, shoot, what's the word? Can't remember it off the top of my head. It's kind of like a clot of protein, right? It doesn't fold properly, and that's very toxic to the cells, and the cells kill themselves. So that's a kind of a classic progressive neurodegeneration disease. Unfortunately, it's a horrible disease to have. I had a family friend who had it, and it's really not a very pleasant thing to experience.
My brother has it. This is
Oh,
why
really?
my
I'm sorry.
kind of interest in it.
Yeah, I
Trying
should
to
have been.
understand it.
Right. So it's rare to see it in younger people because it takes time for that threshold of repeat numbers to be exceeded to the point where it starts becoming a problem. But if you inherit like essentially a gene which has a number of repeats that's very close to that limit, it's not going to take as many rounds of DNA replication to exceed that limit and start seeing problems. So that may be why. I'm assuming your brother is, younger than 40.
Yeah, he has the juvenile onset version of it.
Right. And so then he most likely inherited from one of your parents a Huntington gene with a already existing large cassette, but wasn't large enough to cause disease in one of your parents.
Okay.
It's kind of, how the inheritance of that would work for juvenile onset. For others, it just takes longer to exceed that limit, and so you tend to see in people that are 50, 60 years old and older. But the general, in terms of cures, scientists are not super well up on Huntington's disease. It's not something that I've spent a lot of time studying, but essentially helping the cells deal with that. Damn it, I wish I could remember the word. Sometimes things just leave my head. Kind of helping cells get rid of those protein agglomerations more efficiently will basically delay the progression of that disease, because it's really it's an intrinsic response to protein stress that causes those neurons to die, and those neurons dying is what causes the motor difficulties that people with Huntington's experience. So there is hope for sure, and lots of people much smarter than I am are working on it. So hopefully within his lifetime there will be drugs that will help that process, delay it, allow him to have a good quality of life. So anyway, awesome question. Yeah, and I'm very sorry to hear that it's affecting your brother too. That's not a very pleasant thing to have to deal with. So quickly moving on, we've got another 10 minutes left. So what I want to do is just finish up by highlighting the fact that RNAs are not simple molecules. RNAs are actually one of the most fascinating molecules in biology, if one were to pick one, right? And the reason for that is that they share many of the properties of DNA, but they can do a lot more than DNA can do, right? So they can base pair with themselves. They can form really quite complex structures. tRNAs actually are a good example of that. But what you can see here is that it's kind of a little bit hard to see, but these two different parts of the same molecule are base pairing, right, into a series of basically a stretch of double-stranded RNA, right? They can also move around, they can flex, they can do all kinds of funky stuff, right? So RNA has often been thought of as like the uninteresting intermediate between DNA and protein being the sexy one. But RNAs are actually a lot more interesting than first thought of. And so actually with Rose's question, we got a little bit into kind of mutation and genetic basis of disease, right? So one thing that is really important to stress, and this is actually a common misunderstanding, right, amongst biology students in general. The only source of novelty, genetically speaking, is mutation. That is it, right? So all of why I look the way I am, the way I have, like, well, thankfully, I have like 8 fingers and 2 thumbs, right? Why I have despite my best efforts recently, why I'm bilaterally symmetrical, right? I have an ear on both sides of my head, not just on one of them. All that kind of stuff. That is the outcome of millions of years of accumulating mutations. Not me personally, right? Because I'm only, I'm not millions of years old, but in my genetic lineage, on our genetic lineage, right? So DNA replication causes mutations. Cell division does not cause mutation, other than the fact that DNA replication can cause mutations. All kinds of things can cause mutations. DNA replication is a common one, right? There's intrinsic insults or causes of mutations. There's external ones or extrinsic ones, such as, smoking, for example. Generally, really, really bad idea. Like, why people pay money to smell bad, I don't know. Oh, sorry, I'm getting my personal opinions in here. I shouldn't be trying to tell you whether you should or shouldn't smoke. But in terms of genetics, the chemicals that you generate by smoking do cause mutations, right? Just the same as when you go outside sunbathing, UV can cause mutations in skin stem cells. If you go for a swim in a pool for nuclear waste, that'll also cause a bunch of mutations. But it doesn't, the fact that mutations occur doesn't necessarily tell you what effect they're going to have, right? So one thing may be that, well, the general point to make is mutation is random, right? There's no guiding hand or susceptibility, let's say, to mutation, other than what we're talking about with polyQ genes or trinucleotide repeat diseases, right? So typically, almost always, mutation is random, and quite often it's what we call neutral, which means it doesn't have any effect, right? So a classic example of a neutral mutation will be one that's in some part of your genome that doesn't encode anything. Doesn't matter if it changes, it doesn't do anything. So you just kind of pass that on to your offspring and they pass it on to theirs, and it doesn't make any difference, right? That's the majority of mutations. As I was saying earlier on, you can have mutations occur in the third base of many codons. That's natural in a protein coding gene. They'll also be neutral, right? Because they don't do anything, right? So those are called silent mutations. So that's the majority. Of the rest of mutations and their effects, most of our important point is that's a heritable change as well. And we'll get, there are other heritable changes in DNA as well. Most of them will be deleterious or bad, right? So they will reduce the function of the protein encoded by that gene in some way or other, right? So for example, people that have red hair, it's typically because they have mutation in a gene which encodes a receptor for melanin, or melanocortin, sorry, which is a hormone that tells your cells to produce melanin. So they don't, so they have red hair, which is kind of cool, right? But it's still a deleterious mutation, right? Because that protein isn't functioning the way it should. And very rarely those mutations may enhance gene function or the fitness of the organism, right? And it's kind of worth pointing out that, say, I've got a mutation occurs in me, right? Actually, I'm a terrible example because I've finished having kids. Say a mutation occurs in one of my children. Where does that mutation have to occur for that to be inherited?
Genetically?
Yeah, it has to be in the genome for sure, right? So, but let's put it this way: what part of their body must that mutation occur in for it to be inherited? This is like a completely new mutation.
Wouldn't it be in the chromosome?
Yeah, it would also have to be in the chromosomes for sure. But we have chromosomes in all of our cells, right? What particular cells would this mutation have to be in? It'd have to be in a gamete, or it'd have to be in a germ cell, right? Some part of our reproductive system that will be used to make future generation, right? And so even if, I went outside and, I put my pasty white British skin in under the sun and I got a whole bunch of mutations, none of those are going to live beyond me, right? Whatever those mutations may cause in terms of phenotypes, right? So for a mutation to be inherited, that mutation has to be in the germline, right? And so for a start, most mutations are not going to be inherited. The ones that do, even if my daughter did have a mutation in her germline, right, and that mutation was actually passed into a new individual, right, which is a kind of low odds given the number of gametes that we all have, that's still one copy of that mutation in the whole of the population, right? So its frequency is tiny, right? And so it takes a very long time and a lot of selective pressure for that one mutation to rise to a level where you start to see it in a population, right? It has to confer some kind of benefit, right? So the majority of mutations that occur disappear within that generation. And even ones that do get passed on to a new generation, they typically won't increase in the population unless they confer some kind of benefit.
I actually,
Yeah, go
sorry.
for it.
I have a quick question.
Mhm.
I know it was mentioned that usually the mutations are either bad or neutral, and there's some examples of the the bad mutations. But I was just curious if you had any examples of mutations that increase fitness.
Oh yeah, loads. A really great one, which occurred about 6,000 years ago, is tolerance of lactose. It's a really cool one in our kind of human history. So majority of people of Asian descent are lactose intolerant, right? They can't drink cow's milk, right? And then around 6,000 years ago in the Eastern Mediterranean, there's mutation that appeared, which for whatever reason, perhaps a change in an enzyme like lactase, right, that would digest lactose, that appeared. And so that allowed people, rather than, people would raise cows, obviously, but cows are fairly non-divisible, right? You can't eat half of a cow and then keep the rest of the cow kind of grazing in the pasture. And so after that point, people were able to raise cows for milking. And so not only did you get the cow at the end, for steaks and barbecues and stuff, or whatever you did 6,000 years ago, and hadn't made it to Texas yet, I guess. You were able to harvest the milk as well. And so that was actually thought to be one of the reasons why societies got more stable and richer in Europe over the
Pretty
years
cool.
and less so elsewhere. So
Okay.
I mean, it's kind of, they're hard to think about because we kind of view us as we are, but we are the result of thousands and tens of thousands of those changes, right? So they are caused by something, right? It's just a little bit hard to think about what they're caused by. But yeah,
Cool. Thank you.
cool question.
Yeah.
All right, so I'm not going to go through this list at the end because A, we've run out of time, and B, you can read as well as I can. But essentially this kind of gives you a general overview of the types of mutations and how we can think about them, right? And it depends on what kind of question or perspective you have, how you would classify a particular mutation, right? Whether you're talking about at, you know, the molecular level, or if you're talking about what causes it or what the consequences are, for example. And so that's worth a quick read through, and that will give you some kind of more context, essentially. All right, everybody, that was a lot of fun. I hope you enjoyed that too. If you have any questions from today's class, write them down, bring them with you on Tuesday, virtually speaking. And I'm more than happy to spend like the first 10, 15 minutes going through any of those questions. All right, thank you very much, everybody. I shall see you next week. Have a great weekend.
Thank you. Have a good weekend.
You too.